Dora mentioned, my lab is interested in understanding how multiple organs communicate information about nutritional status to each other. And we're focused on two very particular tissues that I will get to. But kind of thinking about it, like taking a step back, organisms ranging from insects to humans are going to be experiencing differences in nutritional input across a variety of time scales, right? So one day you might eat really healthy food, and one day you might not eat so much, healthy food. One day you may have only eaten lunch, right? Because you've been so busy, and another day you may have been snacking all day. And so each individual organisms are composed of multiple tissues, and all of those tissues are going to have to respond appropriately to nutritional input, right? And coordinate that response so the whole organism can, do what they need, do what it needs to do. And so that's true for humans, that's true for fruit flies. And there are a variety of reasons why I use fruit flies as a model system. But one, it has many of the different tissue systems that we might find in humans. So there's a central, let me get my little pointer. There's a central nervous system here in green. It's got a muscular muscle system shown here in red. It's got reproductive tissues, females have ovaries, males have testes, fat tissue, gut, all of that stuff. And many of the genes that are found in humans that are known to control these nutritional responses at the cellular level are also highly conserved in Drosophila. And we can use the awesome power of Drosophila, genetic power of Drosophila, to manipulate gene expression, make mutants, overexpress things in a tissue-specific way. So right now, our major focus is trying to understand how fat tissue, and insects and fruit flies do have fat tissue, how fat tissue in the flies senses nutritional input, and we're focusing specifically on nutrient sensing pathways like insulin signaling, how they function in the fat to communicate to the ovary. And in fruit flies, the ovary is a stem cell supported tissue. And so that's where our interest in adult stem cell populations comes into play. The Drosophila fat here is depicted in kind of all of this brown stuff here. And it has two major cell types. The adipocytes or fat cells, which are analogous to fat cells in white fat tissue in humans, and the enocytes, which are liver-like cells. We can, using Drosophila genetics, we can manipulate gene expression very specifically in these cells. And so here is an example of that. And to spare you any of the nitty-gritty details, what I'm showing are these are actual pictures of fat cells from flies. And actually this red strip here are those enocytes that I've shown up here. And in this picture, you see that all of the fat cells are lighting up with green or green fluorescent protein. And that's in this single channel image here on the right. And we have the ability to control gene expression only in these fat cells. And so what we do, many of our studies, we knock down components of the insulin pathway, like the insulin receptor, in those fat cells. And then we measure and see what happens in the ovary or any other tissue. The ovary in Drosophila, here depicted in purple, is a stem cell supported tissue. It's a paired structure, just like the ovary in humans. Now, there are some major differences, but there are some overlapping similarities. But this ovary, we can dissect out the ovary and look at individual units. And so here each of these like little strings are these individual units in the Drosophila ovary. And the stem cells are at the very tip here, kind of where this red region is here at the tip of each of these ovarioles, that's what we call them. And we can get really close and look at these stem cell populations. So if we go focus down even further, if we look at that very anterior most region of those ovarioles or egg producing units, we can see stem cells. And so I'm outlining here, there's two of them shown here, 1 and 2. And so what you might be guessing from what I'm showing you is that my lab uses lots of genetics, but also microscopy. And so we're looking at the stem cell populations, at their progeny and what happens. There are kind of right now two major branches in my lab. And so when I originally started, we had very strong interest in understanding how nutrient sensing pathways like insulin signaling, TOR-mediated signaling, the amino acid response pathway, how they function in the fat tissue to communicate to the ovary to regulate egg production. And so we do lots of that. But now we have some newer projects that are interested in understanding how obesogenic diets or high fat and high sugar diets influence the way that the fat tissue functions, influences how the way that the ovary functions, and also maybe the way that they communicate with each other. And here is where our interest in aging comes into play, where we, many of the projects that I have for the students that are ADAR, as ADAR students in the South Carolina program, trying to understand the interaction between obesity or obesogenic diets and aging, right? So do high fat, high sugar diets exacerbate any of the aging phenotypes that we might see in adipose or ovarian tissue function? And one thing that I forgot to mention is that, so I originally came to be a part of the ADAR program as a mentor to some students. And then I've recently, or over this past year, become like a staff member and helping to think about how we implement the South Carolina ADAR program. And so now I'm going to switch gears, and I'm not going to talk any more in detail about the science, but I'm happy to, if anybody has any questions, either after we talk here or after, in the coming months, years, whatever. But now I'm going to switch gears and talk about a little bit like, how did I end up here as an assistant professor? And I will say, let's see, I think there's a chat. Oops, my pointer, hold on. Let me take off my pointer so that I can. Does anybody see what the chat says? Let's see.
Yeah, we can.
Okay, thank you, Seth. Oh, sorry, I thought there might be a question. Thank you. I love my system too. So
if there's any questions, I'll stop you to ask you, or at the end, I can ask you whatever you prefer.
Yeah, anyway, right? We can stop. It's fine. So I will say that as an undergraduate student, I didn't necessarily think about, being a faculty member as a thing that I wanted to do. I was always interested in science and math from a little girl, right, until all through my academic career. And you'll see in a second here that I ended up, I really, really do enjoy biology. So the title of this slide says, stay open-minded and don't be afraid to change your mind. And what I'm going to do is just kind of walk you through here how many times I changed my mind about the things that I was interested in. So I did my bachelor's at the University of Virginia. I'm from Virginia. And actually from when I was like a fourth grader, I thought I wanted to be an astronomer. I had this awesome science teacher who, when she taught the astronomy lessons, she was so enthusiastic that I really, really thought that that's what I wanted to do. And I was excited about it all the way up from fourth grade, all the way up to college, right? My first year, except when I got in that first year and I took astronomy. Yeah, it was not good. It was, the stuff was interesting. The material was interesting. It was just a transition from being a high school student to an undergraduate student. And I had was not smart. I had 3 classes back to back on Tuesdays. So 3 long classes on Tuesdays. The astronomy class, I believe, was either the first, so it was early in the morning. And basically I didn't do well. And I didn't pay much attention, and I did not do well in astronomy. So I said, you know what? I don't think that's for me. But I still always had an interest in science and math. And so my next kind of thing, I actually declared, or was about to declare, a math major. And I took I had some credits from AP credits for stuff. So I started off, not at the basic math, but I took calculus, did all that. That was fine. But then I think it's ordinary differential equations, one of those weed out classes. Yep, it weeded me out. And I realized that I did not want, it was fine. I understood it, but I just couldn't see me going beyond that particular course. But so all the while, though, I was taking biology classes, and I was taking these labs, which surprisingly enough, even for the intro labs, I really enjoyed. I enjoyed those labs. I was doing well in the biology classes. And, given how I felt now about astronomy and math, and it was time for me to declare a major, I declared biology as my major. And if any of you are biology majors or have been biology majors, most of the time, if you tell people you're a biology major, they ask you, oh, are you pre-med? Or they may assume that you're pre-med. And I was never, ever pre-med. I never wanted to go into medicine. And so when I got asked that a lot and got kind of blank stares when I said, no, I was not going into medicine. And I really had to think about, well, what am I? What am I going to do? So all the while I was taking biology classes, as I got up, past all of the entry level things, all of the core requirements, I was taking some higher level biology classes, including labs. And I took a developmental biology lab, which I absolutely loved. And then I took another kind of more in-depth developmental biology lab where we worked with all different types of model organisms. Fruit flies, mice, sea urchin, frogs, chicks, everything. And we had to write a grant and all this stuff for the course. And I really enjoyed it. And so that's when I decided to, I think I like this kind of research aspect. I was at my last year, my last semester. I had taken everything I needed to take. So I needed to fill credits because I was on a scholarship and I had to have a minimum number of credit hours. And I was just looking through the course catalog to figure out what to take. And I saw this independent research. And so I was like, you know what, well, maybe I'll do that. And I asked the postdoc who happened to be teaching the developmental biology lab if she would allow me to do research under her supervision. And she did. And that's where I did some work with Xenopus. And I was like exposing Xenopus eggs to phenylchloroform, which, wasn't much of a real project, but I was in a lab and I was working with frog eggs and all this stuff. And I absolutely loved it. So from there, when thinking about what I wanted to do, I realized I want to keep going on with this research. But as you may have noticed that my grades my grades at my first year were not great. And I was taking chemistry too, which is not my strong suit at all. So, I didn't feel prepared to go into a PhD program from undergraduate. And I'm from Virginia. College of William and Mary is in Williamsburg, Virginia, which is actually just 30 minutes from my hometown. And I said, you know what, let me think about a master's program to really see if this is what I want to do. And I can be closer to home and that kind of things. And if I need to have another job, I can do that. So I did a master's program at William and Mary where I was looking at sperm egg fertilization in the nematode worm Caenorhabditis elegans. That's where I learned all of my microscopy, love microscopy. And I just kind of, that was a great training experience there. It's a small liberal arts school. So, and my advisor was teaching and in the lab. And I just loved that she had both of those aspects to her career. So she encouraged me to, she was like, if you really want to continue on, you have to, and you want to be a professor and you want to continue to do research and to teach, then, you're probably going to do a PhD and a postdoc and then think about an assistant professor position. And so she was actually a postdoc at the Carnegie, which is associated with Johns Hopkins, the Homewood campus. And so she encouraged me to apply to that program. Well, I think we're going to get to imposter syndrome at some point, but based on, my academic performance early on during my undergraduate career, when she encouraged me to apply to Johns Hopkins, I was like, there's no way they're going to accept me. So I applied to 4 different schools, UNC, Chapel Hill, Duke, Rutgers, and Hopkins. And I got interviews at all of those places. And when I went to UNC to interview, loved it there. The other ones I didn't was kind of iffy about. They were good programs, but didn't have a strong feeling. And I almost didn't go to the Hopkins interview because it snowed. It had snowed right before. I was like, I already really want to go to UNC. I don't want to travel. And, there's no point. my advisor at William and Mary, she was like, it's, it's a free trip. If nothing else, it's a free trip. You really should go. So I went, and turns out that I loved it, right? They had a lot of developmental biology labs, both at Hopkins and at the Carnegie Institution, the Embryology Institution. And so, it was a really great fit, and I would probably have more opportunity to find a lab that fit my developmental biology research interests. So, okay, ended up being accepted and you decided to go there as a PhD student. While I was there, I rotated in three labs, a fly lab, actually 4 labs, a fly lab, a zebrafish lab, and 2 mouse labs. And if you would have asked me before any of this, would I ever rotate in a mouse lab or work with mice? I would have told you absolutely not. Did not ever want to interact with mice, didn't want to work. So here kind of you'll see changing my mind, maybe not, not thinking about certain things for myself. So I really enjoyed the zebrafish lab. But then I was kind of not forced to, but by way of ranking and how students could get into their rotations, was put into a mouse lab. Well, I loved it. I enjoyed the research in that lab. And while it did take me some getting used to working with mice as a graduate student, when you have to do the mouse work by yourself, you get over your hesitation really quickly. So I worked with the mouse lab. I was doing its developmental neuroscience. I was looking at how the sympathetic nervous system develops. I had a mouse mutant that had defects in the sympathetic nervous system. And so I really, really enjoyed that project. And then when I decided to, and all the while I was thinking that I wanted to be, continue as in academia, assistant professor, do research and do teaching. But I really knew that I wanted to be not at, and this, it may sound strange coming from based on where I am now, but not a only research focused institution, because I really enjoy training students. I trained students when I was a master's student, undergraduate students, and I enjoy teaching. So I wanted to be able to get to a place that would allow me to do that. And I didn't think that mouse studies would be conducive to the things that I wanted to do, like, forever. Maybe I don't even know. Maybe I'll work on mice at some point again, but it wasn't the model system that I wanted to keep up with for a variety of reasons. But I was still interested in how does the environment influence reproduction and development. And so I just kind of Googled different universities or different labs that were doing that type of research, but I was restricted to the East Coast. So I was kind of focusing on schools in the East Coast. And actually the school, Hopkins Bloomberg School of Public Health, had a lab that really, really fit for my interest. And that was focused using fruit flies. And kind of, this is where I developed the studies that I'm currently working on now. And then kind of to bring it all back, as I was a postdoc finishing up these studies, that's when I really started to think about places where I wanted to be long-term. And all of the places that I applied to either were teaching heavy or teaching plus some research. So while I am at the University of South Carolina, which is it's classified as a research intensive university, there's a big undergraduate population, lots of opportunities to teach and to train students in the lab. And so that's where I've kind of focused my efforts in all of my job applications, and was able to, identify here where I am now, the biological sciences department, as a place that would be a good fit for what I'm currently working on. Any questions? No? Questions?
Thank you. Can I start off with a question?
Yes. Mm-hmm.
So how did your family feel about this progression of astronomy to math to biology to professor?
Yes. So, and I'll stop sharing now. So when my interest in astronomy for this very long time, from like literally 4th grade until my first year of college, every time when people would ask my parents, like, oh, what's she interested in? I would tell people what I was interested in. They'd have this look like, I don't know. Like, I don't know. But they're like, she wants to be an astronomer. She said that, you know, she's been saying this for a long time. So we believe that she will be, right? And I will say that my parents, like, I'm so thankful for, like, it doesn't matter like how out of the box anything I said I wanted to do was. They were always like, we don't understand it, but if that's what you want to do, that's what you want to do, and we're gonna, you know, help you do it. So you know, that they were totally down with that. With kind of thinking about biology and wanted to do research, yeah, they didn't understand what it meant, but again, they were just like, well, if that's what you want to do, and you know, that's what you should do. And now, even now, they ask me to, like, okay, write down what you study, so that, and write it down and give it to us, like, on a piece of paper. So, but they tell everybody, they tell everybody what I do, so they, you know, are able to explain what I do, the research that I do, that I'm a professor and I have a research lab. And so they're like super proud of it and have been supportive all the way.